I'm also working with researchers from the site, trying to make translational and research program in very specific subtypes of AML. So we have had a focus on IDH mutant enzymes in these cells. And first we started with preclinical models, and thereafter we were the first in Europe and with our colleagues in the US to use the inhibitors IDH2 and IDH1 in phase 1 clinical trials. The fact that we have had translational research using these molecules prior starting the trials helped us to better understand how the drug could work, and that was very fruitful because we were able to show that the drug is a differentiating agent. This is not a cytotoxic agent at all. So we anticipated one complication associated with the use of the drug, which is the differentiation syndromes that can be seen in IDH1 and IDH2 mutant enzyme with the use of these compounds. The other thing, we thought that it could take time to achieve a complete remission because this is not a cytotoxic agent. It takes time to revert the hypermethylated phenotype. So that the administration of the drugs, of these drugs, requires time. The first response are seen after, in general, 2 cycles, and best response after 5 or 6 cycles. A cycle is defined by 28 days. So response rates are very high in that heavily pretreated population. The IDH2 inhibitor, Enasidenib, has been approved by FDA recently based on the phase 1, phase 2 trial, which is rather rare. Just to remind that these mutations have been first described in 2009, and the clinical trial started in 2013. So that's very, very rapid, and registered in 2017. So this is really very interesting.